Okay, thanks for the opportunity to talk today on a project that's really emerged over the last couple of years as we've come to understand aging biology in a little bit more detail as it pertains to cancer. So, colorectal cancer is a disease that is a big focus of my research, and it's the fourth most common cancer in Australia. There's about 15,000 annual diagnoses of colorectal cancer each year, and it's actually the second deadliest cancer behind lung cancer, with 5,000 unfortunately succumbing to the disease. We know that age is a really significant risk factor for colorectal cancer development. So you can see here that there's really low risk of developing colorectal cancer in the first 3 or 4 decades of life, but we have an inflection point that occurs around age 30, and then an exponential growth in the incidence rate of colorectal cancer going all the way up into the 80s. Some of you may be familiar that this landscape is changing somewhat. So here we've got depicted different age groups and the age standardized rate. Colorectal cancer is notionally considered a disease of aging, and that was the case for many, many decades, with incidence either remaining flat or decreasing in the younger age groups. But what you can see over here is that there's been this troubling rise in the incidence of the disease amongst younger patients. And this has been noticed across a lot of Western countries throughout the world. And you might have seen recently at AACR, there was a poster presentation there that gained some media attention, showing that a lot of these early onset cancers are associated with an accelerated aging phenotype. So it's potentially coalescing around that idea that aging is a risk factor and that it is potentially modifiable by environmental influences. So colorectal cancer isn't just one entity. So we have a number of different pathways that can actually lead to colorectal cancer developing. So the one that we typically think about is the conventional pathway, and that's usually initiated by mutations in the Wnt signaling axes. This is usually a mutation of the APC tumor suppressor, which leads to constitutive activation of that pathway. But we can also get the alternative pathway, or the serrated pathway, and this is triggered by a MAP kinase mutation, and usually a point mutation in the BRAF proto-oncogene, which leads to, among other things, the development of a hypermethylated phenotype, an epigenetic phenotype, and the accumulation of microsatellite instability, which is where a whole heap of mutations occur throughout the genome. So like I mentioned before, there's this real coalescence of aging biology and cancer that we've come to appreciate over the last 3 or 4 years. And this is from a hallmark paper in Cell Metabolism by Oden and colleagues, looking at what they call the meta-hallmarks of aging and cancer. So aging, if you could look at it at a fundamental level, is a process by which the body is trying to protect itself. At a cellular level, it's trying to prevent damage from being passed on to further progenitors and into causing diseases. So we have these meta hallmarks. So these are things that are common in aging and cancer. So we know that there's an accumulation of genomic instability, epigenetic alteration, dysbiosis, and chronic inflammation. But we also know that there's a number of processes that are designed to do what I mentioned before and antagonize cancer development, such as stem cell exhaustion, telomere attrition, and replicative sort of insufficiency. So there's this tug of war going on between the things that go on with aging that should be preventing cancer and those that might actually be promoting it. And just to give a sort of case study about one of these meta hallmarks, we've looked at epigenetic alterations in colorectal cancer for many years now. And this is just a study from that we published in 2019 showing that you do really get an accumulation of DNA methylation in the promoter regions of cancers. And you can see here that the cancers with the most methylation tend to be those with that BRAF mutation that I mentioned before. Intriguingly, we also see an association with the age of diagnoses and how much DNA methylation we're identifying in that cancer, with those hypermethylated ones occurring at an average age approximately 77 years. So we then went on to do a study where we looked at this in more detail using a mouse model where we induced the BRAF mutation. And what we saw is that a lot of the DNA methylation alterations that would usually occur during aging are substantially accelerated in the presence of that mutant BRAF. So it seems again that there's this coalescence or this synergistic activity between the oncogene and a normal aging process. And importantly, when we look at what happens when we induce that oncogene at different time points, we can see that the oncogene isn't very potent when you induce it at weaning, but when you induce it at 9 months after a period of normal aging, we see 40% of those animals going on to develop cancer. So something is going on here to lead to the aging process increasing a basal risk of cancer. And there's been a number of other studies that have identified similar phenomena. So a study published in Nature a few years ago looked at regenerative capability of organoids derived from crypts of aged individuals. And what you see is that if you try and derive an organoid from a young individual, you will derive many more organoids per crypt than you will from an aged individual. And you can see that depicted over here as well. They identified this molecule called notum, which is an inhibitor of Wnt signaling that's overexpressed in the aged intestine. And that they identified is a key inhibitor of regeneration and of this sort of ability to form organoids in the epithelium. A study by one of my colleagues down at Monash while he was in Scotland, Dustin Flanagan, looked at the same molecule in cancer. And what they found is that if you have a loss of notum in these APC mutant animals, you won't be able to form lesions and fix lesions as effectively. And moreover, when you knock out notum, this gene that goes up with age in Billen-Cree, ERT, and APC min mice, you see a profound difference in the survival of these animals. So indicating that this age-associated molecule is actually important in the development of neoplasia. So there, it naturally follows that addressing aging as a risk factor for colorectal cancer could identify novel preventative therapies. And that begs the question, can we reverse aging? So this is something that one of my colleagues at Monash also looked at recently in a MPJ Regenerative Medicine publication. And they identified a fairly similar phenomena that if you isolate intestinal stem cells from young mice, they'll grow significantly better than if you isolate them from old mice. And you can see that depicted here with the young animals forming organoids much better than those that were aged to 22 months. They identified a number of different transcription factors that are responsible for the transcriptional rewiring that's occurring between those stem cells in the young animals and the old animals. And when they overexpress these transcription factors in those elderly animals, in the organoids, sorry, you get a restoration of that ability to form organoids and that regenerative capability. So that brings me to my Cure Cancer project. So I hypothesized that cellular reprogramming to rejuvenate the aged intestinal epithelium will prevent neoplastic transformation by creating a more competitive environment that lesion needs to compete against in order to form a cancer. So in this project, we're going to use BRAF-inducible organoids as models for age-associated colorectal cancer. These are derived from the BRAF V637 Cree ERT2 model. And as I showed earlier, it's a nice model for age-associated cancer. This is what these organoids look like, and these are derived from aged animals. And you can see over here, you get the development of this gross dysplasia when you activate the BRAF mutation. This tends to only occur in the organoids that are derived from the elderly animals. To date, we've collected specimens from 12 mice and derived organoids. So these are from 2-month-old animals and 20-month-old animals. And our plan here is to use this multi-cistronic vector, which contains those 3 transcription factors that I mentioned earlier, to rejuvenate those elderly organoids and then activate the BRAF mutation to see whether we can have those organoids resist transformation. And we're going to do this by reprogramming a series of different settings and doing an organoid mixing experiment where we'll put in the organoids that have been reprogrammed and their age-matched control organoids that haven't been reprogrammed, and then monitor dominant populations and perform RNA-seq and proteomic profiling. And here's just an image of what that competition type assay would look like. So here we're really trying to see whether those reprogrammed mutant organoids can outcompete those that haven't been reprogrammed and have that aged transcriptional setting. We also plan to take this into an in vivo setting. And to do this, we're going to use an injury model where we're going to induce injury in the colon of mice using EDTA, and then use an intradermal brush to further exacerbate that injury and inject the organoids from this mixed population. And in doing this, we hope to see the engraftment of our organoids in a mixed setting to see whether we can identify those sort of clonal crypt dynamics that might be influencing transformation. So this project is obviously ongoing. I've moved institutions and we haven't made much progress to date, but I'm really excited about where it goes. And what's left is just to acknowledge all the people that were involved in the project and of course Cure Cancer for their generous funding of the work. Thank you.